Hi, everyone, and welcome to episode 108 of Cell and Gene, the podcast. I'm Cell and Gene's chief editor and host of the pod, Erin Harris. In just a minute here, we'll dive into a great story of scientific persistence, clinical innovation, and high precision cell therapy. I'm joined by Nathaniel Fernhoff, co-founder of Orca Bio, a company that spun out of foundational work in Dr. Irv Weissman's lab over a decade ago. Orca has come a long way from an early vision rooted in refining stem cell transplantation to today's late stage clinical breakthroughs. The company's flagship therapy, Orca T, recently delivered strong data from a pivotal phase 3 study showing dramatic reductions in transplant related complications like graft-versus-host disease. Nate and I talk about how Orca leveraged engineering and manufacturing innovation to overcome decades of logistical and biological hurdles. We talk about their commercial strategy as they move toward BLA submission and a whole lot more. Nate effortlessly shares really important information, and I think you'll find him to be just as knowledgeable as I do. And so without further ado, here's my conversation with Orca Bio's Nate Fernhoff. Enjoy. Hi, Nate. Welcome to Cell and Gene, the podcast. Thanks for being here.
Thanks for having me.
Good. So, all right, you co-founded Orca Bio on work that started over a decade ago, probably more, in Dr. Irv Weissman's lab. So talk to us a little bit about what did that journey look like from where you were then to where you are now in terms of commercialization following your positive phase 3 results?
Sure. So if I could take a step back and maybe tell you a little bit about Orca.
Yeah.
To start with. So at Orca, we are aiming to build better, safer cures for patients with blood cancer. If you are a patient today with acute leukemia like AML or ALL or MDS, an allo stem cell transplant is really your best chance for a cure. But there are severe complications that can result from a stem cell transplant. And it's one of these sort of where the cure is worse than the disease for some of these patients. And the patients will have complications like graft-versus-host disease, serious infections, organ failure. And so Orca Bio is a late-stage clinical company, and we're building high-precision cell therapies to target only the therapeutic cells in the graft so that we can really isolate that effect. Ultimately, we're aiming to cure that underlying hematological malignancy, but do it with fewer toxicities and fewer complications. As far as where the company's at in general, we're a late stage clinical company. Our lead candidate, Orca T, has now met its primary endpoint in a pivotal phase 3 study. And we are working very closely with the FDA to make Orca T available to patients and their doctors as quickly as possible. Okay. You asked about the origin story.
Correct.
For Orca. So let me dive in a little bit to that.
Sure.
Pretty interesting story. I'm gonna take you way back to the late 1980s when Irv Weissman, who was already at the time a famous immunologist, was working on bone marrow transplantation, and a really important problem. People had known that bone marrow can regenerate in a recipient. So you can regenerate the hematopoietic system, the immune system, using bulk bone marrow. Irv did something really interesting, and he purified a very rare but potent cell from the bone marrow. And this was, less than a fraction of a percent of the bone marrow. And he showed that the capability to regenerate the entire blood and immune system in the mouse was found in this exceedingly rare cell type, and only this exceedingly rare cell type. Mm-hmm. And what was different about this experiment was that Irv started was one of the first adopters for flow cytometry. And this is the ability to purify the cell to effectively homogeneity, right? So you have a pure cell population, and now you can really understand what it does in the lab in the context of an experiment. Fast forward 15 years, and now Rob Negrin, who is a bone marrow transplanter, also at Stanford, working in a building across the way, is working to find better compositions of cells for a stem cell transplant. And Rob similarly finds that he can purify regulatory T cells to control graft-versus-host disease and really prevent it from occurring in mice. These findings are reproduced in labs worldwide, and people can really robustly prevent GVHD in mice by using purified regulatory T cells. A number of pioneering graft engineers try to adapt this finding to the clinic, and it's very difficult for a number of reasons. Mice have about 100 million cells in their bone marrow. Humans will have about 100 billion cells that you're working with. So it's really, there's a huge, massive difference in the scale that you need to purify. Further complicating matters, the regulatory T cells were shown to perform best when they are not cryopreserved. And so now you've got to get the cells from the donor to your manufacturing facility. You've got to do all of your manufacturing and get it back to the recipient site all within 72 hours. And so a few folks were able to, with heroic efforts in single site, small scale academic clinical trials, they were able to test some of these ideas. But the big idea for Orca was to come in as a manufacturing platform and to try to do this in a more scalable fashion. I think what was really interesting about this for us at the time was that the problem with this approach could be solved with engineering solutions. It wasn't really a biology problem anymore, where you're worried about, is this drug going to work? It's a novel mechanism of action. It's the first in human study. So many people had reproduced various studies with regulatory T cells to show that they could prevent GVHD. This was really a question of, could we build all of the other systems that we needed to do this type of production and logistics so that we could maintain product quality and get it out to every patient who needed it.
In a short amount of time.
In a very short amount of time. That's right. That's right. So we spun Orca out of Stanford with the idea of this manufacturing platform. And we built a really incredible team of scientists and engineers and just generally people who were passionate problem solvers. And as we stabilized the formulation and the manufacturing process to do this for patients, we started engaging with clinicians, folks from around the country. A lot of these early pioneers that I mentioned earlier, the folks who were passionate about graft engineering as a way of reducing transplant complication, and they were invaluable in designing the clinical study and helping it get off the ground in the early days of the company. For Orca T, we treated about 200 patients across all the different studies in early phase clinical trials, where we were learning things about how the drug behaves once you introduce it to a patient. We were learning about the optimal way to deliver it to a patient, things like the conditioning regimen and pharmacological GVHD prophylaxis. We were figuring out how to target all of these things and dial in the best efficacy with the lowest rates of complications. Once we had done that work, we engaged with the FDA and designed a pivotal phase 3 study with them, and that study is now complete. It was a randomized trial comparing Orca T against a standard of care stem cell transplant. We randomized about 200 patients, almost 200 patients, across 19 different centers in the US. So there's a large randomized multi-center study. This is really sort of a gold standard experiment in clinical development. And we've recently announced that this was a positive study, a very positive study. We saw the primary endpoint in this study was survival free from graft versus host disease, which we saw in 78% at 1 year of our Orca T patients, and we only saw in 38% of the patients who received the standard of care allograft. So a really important difference. We also, we saw that overall survival at 1 year in the Orca T arm was 94%, 83% in the standard of care arm. And so we think that there are a lot of benefits. That was not, the overall survival is not statistically significant yet. The study wasn't powered to look at overall survival. But we think these are really promising signals for the potential of Orca T to essentially replace stem cell transplant for these patients.
Yeah, and promising and impressive for sure. That's, certainly and for patients and their caregivers, providing a lot of hope, which is amazing. Orca Bio, before we move on, can you talk about where did the name of the company come from?
Orca Bio? Yeah. So I'm debating whether to use our like form joke here about curing cancer in whales, but no, the Orca moniker comes from the manufacturing technology that we use, and it's one of the abbreviations for one of the homegrown technologies. It's optical retrieval from cell arrays.
Gotcha.
But Orca just kind of has a nice ring to it as well.
It does. It does. All right. I want to talk to you a little bit about how you're defining high precision. So your platform is pretty unique when it comes to high precision cell therapy. And how are you personally, I guess, defining precision, and how does that translate for better outcomes with, like we talked about, patients with AML, ALL, MDS?
Right. So high precision here really boils down to the purity of the cells that are making up the drugs. And really, I'm talking about, with most interest here, about the regulatory T cells that we use. So Tregs are a naturally occurring immune cell that controls inappropriate immune reactions from other T cells and other immune cells. In a conventional transplant, there are Tregs in the allograft. They're exceedingly rare. They're about 1% or less of the total population of cells. They are surrounded in a conventional transplant by, in particular, T cells, which are needed in the context of a transplant, but they present a number of potential complications for the patients as well. In particular, even a single alloreactive T cell clone can go on to mount and sustain a GVHD reaction in the patient for life. Just like our drug is a living material that is meant to durably cure the patient for life. It's meant to regenerate their blood and immune system. So too, the contaminants that go alongside your drug have the same ability to proliferate indefinitely in the patient for life. And so the purity of those regulatory T cells becomes really important. What we do is we, from the allograft, which starts as a very complicated blood product that has dozens of different immune cells, each of them has its own function, we pull out just the stem cells and just the regulatory T cells. And when a patient, in the course of their treatment, they receive cytoreductive conditioning to reduce or eliminate their own immune system, then the patient is sort of a blank slate. We first give the patient these stem cells and these regulatory T cells. That's really important. Essentially, we are priming the patient with an immunotolerant, tolerogenic environment for the new immune system to reconstitute. So the Tregs, when they're introduced into the patient, they home to the lymph node and to the spleen, and they set up shop. They start proliferating. 2 days later, we add back a controlled dose of those T cells. But now those T cells are not exposed to a completely naive environment. They're exposed to an environment that's been primed by the regulatory T cells. In this manner, the T cells are controlled in their ability to react against solid organs that would result in GVHD, but they are still able to prevent infection. They're still able to carry out what we call graft versus leukemia and control disease relapse. And so the T cells can go on to perform their normal functions without causing these pathogenic functions that essentially destroy the recipient's tissue.
Gotcha. Thank you. So I want to talk a little bit about your PDUFA date that's coming up in 2026. A few of our listeners, I'm sure, are anticipating the same thing with PDUFA dates upcoming. And I'd love to hear from your perspective, so you and your team, what are your priorities as you prepare for the potential launch?
Yeah, great question. Well, look, for clarity, we don't yet have a PDUFA date.
Okay.
So we haven't yet submitted our BLA package to FDA. We're planning to do that imminently. And so we'll be working with the agency to get a PDUFA date, like you said, hopefully in 2026. But there are a number of things that we're focused on to prepare for that date and for product launch and to make this drug available to physicians and patients. So there are really three major initiatives that we're focusing on. Manufacturing, making sure that the supply is available, setting up our commercial team, again, to make sure that the drug is available to patients. And the third is continuing our pipeline development for other indications as well. So but maybe we can talk first about the manufacturing and what we're doing to get ready to scale our manufacturing, our GMP manufacturing for commercial operations.
Definitely.
I think the most important thing that we've been working on for the last few years is the setup of the commercial facility. This is really an incredible facility. It's 100,000 square feet with clean rooms and testing labs, warehouse, office space. It's really sort of the crown jewel for the company. And during our clinical trials, we have now produced product for more than 500 patients on our clinical studies. That is a lot of patients, but frankly, pales in comparison to the impact that we can have once we're once we have a commercial product. And so this new site we expect can make at least 3,000 products a year when it's fully up and running. It is currently operational. We are producing product for our clinical studies, the ongoing clinical studies with Orca T that we have in the pipeline. We are making those products out of that facility, and we are very actively growing, particularly the manufacturing and quality teams, to ensure that as we scale this production, we don't compromise product quality. We have to deliver the same high quality product that we've been making. We need to continue making that and make a lot more of it. Mm-hmm. So a lot of work happening with our manufacturing and our tech ops group.
Interestingly, what we talk about this all the time with the subject matter experts that we have on, is when did you begin those conversations / plans? And the answer is always the same, as it should be: early. That's literally always the answer. But when do you, other than early, when did you begin that process?
The exact date that we sort of broke ground escapes me, but very early on in our company's history, we knew that we wanted to own manufacturing. Mm-hmm. These were going to be our patients, and we needed to take responsibility really from the donor vein all the way until we could deliver that to the recipient's hospital. Sure. And so we wanted to do that manufacturing ourselves. We had wonderful partners in Sacramento when we first got started, and we even though the company's research and corporate office is here in Menlo Park, we set up our production facility, first a clinical facility in Sacramento. We learned a lot about how to produce these drugs during the phase 1 trial for Orca T. We also have another trial that we produce our own product for called Orca Q that's treated another 100 patients. we learned a lot during the clinical development about production. And in the lead up to the phase 3, we talked to the FDA about moving to a commercial facility. We wanted to make sure, of course, again, that when you move to a new production site, that you didn't alter fundamentally the quality of your drugs. And so we ran a relatively large experiment during the phase 3 that then allowed us, with regulatory oversight, to move production into the new commercial site. And it's been invaluable that even after the phase 3 has ended, we continue to produce drugs for phase 1 studies and ISTs that we support for Orca T. We continue to gain that manufacturing experience at the commercial site. We don't want, of course, the first day of product launch to be the day that you start production. And so we are constantly producing drug and exercising the system there.
Good, thank you. And I apologize. I know I interrupted you as you were about to carry on with your response.
No, that's great. I prefer it. Please continue to interrupt me.
So anyway, so you had covered manufacturing, and you were gonna, I think, talk about commercialization perhaps, or
yeah. Okay. So we're very excited to set up the commercial organization. This is really entering a new stage of company for Orca. I think to date in clinical development, we did not really grow the commercial function at Orca beyond sort of what was needed from a bare bones perspective to understand the market and understand roughly the road to reimbursement. Mm-hmm. I think now that we have our positive phase 3 data, we are very privileged that we've been able to recruit Steve Gabel, who formerly led the commercialization efforts for Carvykti at Legend. Mm-hmm. And under Steve, we're building out now all of the different commercial groups that are really required to make sure that patients are going to have access to the product. So, we have a market access team that we're building out, a med affairs group, a marketing team, account managers, case managers. So we are very actively recruiting in preparation for launch. I think these cell therapies are different from a lot of other biologics that go out into treat patients, in that we don't have a warehouse sitting somewhere with a bunch of Orca Ts that are waiting on the shelf, right, for distribution. We have to make this as a just-in-time product, and it's specific for each patient. That takes a lot more management and oversight over the logistics as we're moving around these bespoke blood products from one point in the world back to another point, and again, all within 72 hours. And so there are a number of I would say special roles that we are growing and training for here at Orca that can ensure that that happens seamlessly for all the patients that we intend to treat. The pipeline that we're developing at Orca is really important, and this is one of the things that we've done really well, and we expect what's gotten us here is going to get us there into the next stage of company. And we really want to continue to invest in the pipeline. And part of that is figuring out new drugs like Orca Q that may have a different therapeutic effect. And the other part is figuring out who are the other patients that might benefit from an Orca T. I think there's an understanding in stem cell transplant that when you replace somebody's blood and immune system, there are a great number of diseases and disorders that you may be able to cure. Today we use stem cell transplants used in the clinic to cure acute leukemias. It's also used for some patients with non-Hodgkin's lymphoma. It's also used for some patients with sickle cell. The problem with stem cell transplant has never been efficacy for these and other disorders. The problem has always been safety.
Mm-hmm.
And with the Orca products, you have to ask, which of these other indications, how many of these other patients could you treat with curative intent with a safer product? And the answer to that question will come from your clinical development. It will come from, understanding how safe is your product. So, for instance, on the phase 3 clinical study, we saw transplant-related mortality rates of around 15% in the standard of care. That's normal, meaning if you go in for a normal conventional stem cell transplant, there's a 15% chance of death from a non-relapse related cause. It wasn't the original disease, it was something related to the transplant. For Orca T, we saw 3.5%. And that's a big drop. Now, that safety profile for Orca T has been established with myeloablative conditioning. One of the big questions that we want to ask in our pipeline is how safe can we make an Orca T if we reduce that conditioning to a reduced intensity conditioning level, or what's called a non-myeloablative level? This is a really similar question that parallels what people are asking in CAR Ts right now, which is, they believe a lot of toxicity derives from their lymphoablation. So they're looking for other ways to reduce or eliminate lymphoablation in the context of a CAR T. Similarly, for Orca T and Orca Q, our pipeline is now asking questions about how can Orca Q or Orca T be safely deployed with reduced intensity conditioning and non-myeloablative conditioning so that we can maintain durable engraftment and durable cures for these patients, but with reducing that level of toxicity even further. This is really important. So in the US, patients who have AML or MDS or ALL, there are around 50,000 diagnoses a year in the country. Only 7,000 of those patients are going to get a stem cell transplant. And one of the main drivers here is the toxicity of the stem cell transplant. It's the safety of the transplant. These are patients who already, they have the same disease, but their physicians don't think that they're going to be able to tolerate a high intensity intervention like stem cell transplant. So we're really interested at Orca in doing the clinical development for our drugs with these reduced intensity conditionings, with these non-myeloablative conditionings, so that we can reduce even further the toxicities and perhaps make Orca T an appropriate therapy for these other patients.
Mm-hmm.
Once you've established your safety profile in this context, I think there are a lot of other important conditions that for us to start exploring. Things like autoimmune disease, like you mentioned, other genetic diseases, other forms of blood cancer, all may benefit from a durable way to replace the blood and immune system with less and less toxicity. And so, the company has plans to, we had a stage development plan to start looking at these things subsequent this clinical development work that's ongoing now. We had a number of investigators who are already using these products in the myeloablative context, who were very interested, who are aware of the safety profile and thought it was appropriate for some of their patients who have the most severe forms of autoimmune disease, for instance. And they've launched an investigator-sponsored trial that we're supporting in the Orca Q context for primary progressive multiple sclerosis to see if, again, this hypothesis of durably exchanging a diseased blood and immune system for a healthy one, if that can be used to cure these autoimmune diseases. I think, again, folks have used stem cell transplant to address all of these indications in the past. It's just been a safety issue. And with the safety profile Orca Q and Orca T that we're starting to understand better, we think there's going to be potentially much greater impact that we're going to have even beyond hematological malignancy.
Fair. Okay. Before we wrap up, I want to talk a little bit about, just based on you and your tenure at Orca and your entire profession to date, what for the cell and gene therapy biotechs leaders listening, from a best practice perspective, from a lessons learned perspective, we're in a challenging market, whether it's, from an investor perspective, from a, safe and efficacious product perspective. What are some of the things that you've learned along the way that you can pass on to, small and emerging biotechs today trying to navigate a pretty tough environment?
Yeah, it's a good question. Look, I would each of the cell and gene companies is really a snowflake, and there's really not a piece of universal advice that I think I could kind of bestow out that would apply to everyone. Look, the truth is we've had phenomenal investors at Orca, folks who really believe in the mission and the impact that we can have. But I think, as thinking about this question, I would urge people to fail early and fail efficiently. Right? Most ideas that you have are going to be bad, right? And the key is working your way through those bad ideas as quickly as you possibly can. So, as a scientist, if you can get to an answer to a question by reading a paper, you should do that, right? Mm-hmm. Why spend 2 days at the library when you can spend 2 months at the bench, right? This is one of these things that scientists harass each other with. And I think that same lesson really applies in biotech, and it's really about trying to get to an efficient answer as quickly as you can. You got to be your own biggest skeptic and think about where are things going to fail and pressure test things and try to build up the systems as quickly and efficiently as you can that will get you to an answer.
All right, that wraps up this episode featuring Orca Bio's Nate Fernhoff. Nate, thank you so much for being here. This was a ton of very, very valuable information. And like I said, we'll have to have you back on to, I'm really convinced I'm going to figure out this 72-hour, manufacturing discussion so we can really break that down for our audience.
That's great. Yeah, it would be a pleasure.
And thank you, listeners, for tuning in. And we will see you right here next time on Cell and Gene, the podcast. And we will talk to you soon.